20 years ago. So very exciting to be back here. Yeah, Fred talked about my lab. So I do this. If you look in the corner here, you can see this incandescent light bulb that represents me. This is a drawing a logo that my student Bob Smith did many years ago. And so the old incandescent bulb, which is basically an extinct organism right now, that represents me, but my interests lie in two different directions. One is that mayfly on one side, aquatic insects, and the other side is a potato leafhopper, which is a pest of field crops. And my research has been sort of split between those two fields, IPM and aquatic entomology. The nice thing about this project I'm going to describe today is that it combines both sides of that light bulb there. So this is a story. It's a story of discovery, of controversy, of risk assessment, and finally, ultimately, about science just in general. And it's all about if you took this corn right here, standing on the stream bank, and if that corn debris, whoops, goes into let me go back again. Let's see if I can do that. Oops, it doesn't go backwards. So previous, previous. If this corn debris goes into the stream, what is the impact on the insects? And that's what our research has been about for 15 to 20 years now. And so we're getting near the end. There's one part at the very end that we have yet to publish, should be coming out soon, and I'll describe that then. But before I do that, I wanted to mention EntoQuest. Fred mentioned it briefly there. When I was president, we started the EntoQuest weekend. So this is annually hosted by the ESA Eastern Branch in September, and it's been going on since 2022 in September's. It's an informal weekend meeting with hands-on workshops and outdoor recreation activities centered around insects in a natural area. What more fun could that be? And it's a lot of fun. Mostly students, but it also includes professionals as well as hobbyists. So certainly members of the Maryland Entomological Society would be invited to come here. You do have to be an Eastern Branch member. And the one good thing for students who are interested in this, if you have never joined the ESA, we can provide free membership to students in order for them to register for this meeting. So that's where it's been in the past. Now, this coming year in 2026, we're going to meet at Camp Manatawny in Pennsylvania. That's near Reading, Pennsylvania, September 18th to 20th, 2026. There are many types of habitats there to be sampled. This is a typical picture from 2023. You can see it's probably 90% students and about 10% the older people. But we have a lot of different young professionals in here as well. So I hope you can watch out for this. And if you have any questions about EntoQuest, it's on the Eastern Branch website, but also you can contact me, of course, anytime. All right. So what about the risk of transgenic corn in streams? So I'm going to talk about the idea, the controversy about stream ecosystems as background. Then we'll talk about the research for non-target risk overall. And that includes exposure, as well as the effect on individuals, effect on ecosystems, and end with a few comments generally about risk assessment. Most of you, maybe all of you, know about transgenic corn, but just basically to give you some background here, this is a genetically modified organism used to control corn damage caused by European corn borer and corn rootworm. It is the expression of proteins from Bacillus thuringiensis, that's the BT, and we can do that either as single genes or stacked many genes into the crop plant. The cry protein is an endotoxin, so it has to be fed upon by the insect to be effective. The cry protein binds to the gut lining of the target insect, which causes it to stop feeding, ultimately causing death. And as a consequence, it's a very selective toxin. Generally, it's just a few families or maybe an order of insects at a time, and for each of the different genes. I say rising use by producers, but we've sort of maxed out here. But over there in the chart, you can see the purple line is corn and introduced in 1996. It's grown steadily to 2014, shown here, to 80 or not 90%. Galen Dively estimates about 95% of our corn is now BT corn in the state of Maryland. Along the way, in 2016, the National Academy of Sciences developed a report on the experiences and prospects of genetically engineered crops. And they ultimately concluded that it was a safe thing to do. It's safe to the environment, safe to people, and so forth. That hasn't stuck. There is continuing concern for non-target effects. People are concerned about it. And so we do need research to find out if there is any effect or not. So where did I come up with this idea? So I was sampling in this particular stream. This is an ag stream at one of our research and education centers near Clarksville. Um, and I was collecting insects on that fall day it was shortly after corn harvest, and there were corn leaves blowing around on a blustery day. And some of those leaves are going to end up in the stream. And so I wondered, does BT corn affect streams? And so I went back to the office from here, looked it up online, and found out the only aquatic organism tested for BT corn was Daphnia magna, which, as you know, does not feed on corn leaves, does not even occur in streams, is not an insect. And so it certainly wasn't a good answer to whether or not there's any effect or not. So that started us down a pathway of doing research on this question. And I teamed up with Galen Dively and Cerruti Hooks in entomology here at Maryland, but also Chris Swan at UMBC to look at the stream ecology aspects. And later on, we got involved with Chin Wang, who works on food safety concerns in at the University of Maryland. So together, we started collecting data to look at this issue. So, but let's think about this generally. Are streams at risk? Well, certainly streams are pretty places, and they are valued to us for a lot of different things in terms of just water supply for us. We know that in terms of the stream ecosystem, leaf litter is very important. So in this chart from Hall et al. 2001 that shows the annual production rate is directly related to the leaf litter standing crop. We know that that leaf litter is the source of the major energy in many of the small streams. Streams are dependent upon allochthonous energy from the outside, such as leaf litter. We also know the importance of headwater streams. We often emphasize large rivers and so forth, but in fact, these headwater streams are very numerous and they're all over the landscape. It's where there's a lot of processing of organic debris. There's nutrient processing as well. They harbor diverse and unique species. And in general, they are an important transition zone for the exchange and transformation materials at land-water interface. So these are important places in our habitats. And if you consider the river continuums concept that shows us the role of shredders, especially in the headwaters. So this chart shows at the headwaters here, if you look at the proportion of shredders among the insects, it's very high. These shredders are using that leaf input that goes into the stream during the fall and the winter. The leaves are colonized by fungi and bacteria. That begins the breakdown process. And then the shredder invertebrates then feed on that matrix of the leaf material and the microbes. The microbes are what they're really after. They call it peanut butter on a cracker because if the leaves are like crackers, they're really of no nutritional value, but those microbes are of great value. So they're like the peanut butter for the insects that feed on them. Of course, there are a lot of specialized biota associated with streams that could be impacted by not these genetically modified organisms, but especially we were concentrating on the caddisflies. And you all know that caddisflies are very closely related to Lepidoptera. They are sister orders, if you will. And a lot of diversity in caddisflies overall, but some of them are especially good shredders, feeding on leaf material. And one of those is that one on the left there, Pycnopsyche in the family Limnophilidae, common around in Maryland, and does like to feed on leaf material in particular. So that was one of our things that we wanted to look at to see if there's any impact on that caddisfly. And from the beginning, we knew that there was a potential for controversy. If you think of BT as a selective toxin, if you will, if you put that into a stream, there could be harm, and people would be concerned about that. And we certainly knew about that at the time of the start of this research. It was the same time that John Losey fed monarch caterpillars with BT pollen and found them to die. And not too surprisingly that they did that. And that caused an uproar in the among the environmentalist societies. Uh, and so that led to a lot of research. And what that research showed that although there was an effect, there was not any exposure, that those caterpillars were never in a habitat where they would feed on BT pollen in any great amount. And so there was really no impact on monarch populations caused by BT corn from that work. Now, of course, monarchs are a concern in terms of their decline, but there are a lot of other causes for that now. And so anyway, we knew that there was controversy around GMOs, and so we wanted to focus on the science of risk assessment and specifically on the science, not on whether or not it is negative, whether it is positive. We wanted to find out what is the result. Um, and as that was reinforced when a paper came out shortly after we started our work. This came out in PNAS by a group out of Notre Dame entitled Toxins in Transgenic Crop Byproducts May Affect Headwater Stream Ecosystems. Clearly, this is a true statement because it says may, but nevertheless, it leads to the conclusion that there are issues with BT corn in streams. They present data. The science was questionable. There was a lot of letters to the editor and so forth, which you can debate if you want to. But here is my problem with that article more than anything else. The last sentence of the abstract says, widespread planting of BT crops has unexpected ecosystem scale consequences. None of their research led to me to make that kind of a conclusion from that research. So it sort of fanned the flame, if you will, on this issue. So we wanted to really focus on the science. What was the science? The science says that environmental risk assessment is a function of exposure and effect. If there's no exposure, then it doesn't matter whether there's an effect or not. But if you do have exposure, you need to find out if there's an effect on individuals, and you need to find out if there's an effect at the ecosystem level. And so that was the direction that our research headed. We asked for the exposure. We asked 5 major questions here, and I'm going to go through these one by one here. And most of this has been published overall, but we'll start with are streams adjacent to cornfields? This may seem like a silly question, but we wanted to answer it anyway. I got my graduate student at the time, Dilip Venigable, to help me with this because he was able to use GIS to look at this. And I was giving a talk at University of Cincinnati, so he looked at 3 counties just north of Cincinnati to do this work. And here's what he did. He was able with GIS to identify all of the cornfields in a county by their spatial arrangement. And he was able to coincide with that, the streams as well. And then he could query the system. How many of those streams are within 10 feet of a cornfield? Now, this is clearly an agricultural area, so not too surprising that when he did that query, then here shown in red are all the streams that are associated with cornfields. And so when he did this over this whole three county area, and this down to the south side, you can see this is approaching Cincinnati, so it's a much more suburban area. But we see that there is a total length of stream network in all three counties of about 3,800 kilometers. The total length with some portion of the stream within 10 feet of corn, nearly 2,400 kilometers. So we found 62% of the stream length is associated with cornfields. And this is not unlike other measurements we took in other states as well, in other kinds of regions, that certainly cornfields are ubiquitous in our landscape, and they could be a source of input to the adjacent streams. Okay, the second question is corn tissue entering the stream after harvest? And we did that. We used traps to catch the debris, leaf debris moving in and out along the stream edge and so forth. And this shows 2 years of our data on lateral input. The interesting thing was that corn leaves at harvest tend to be heavy because they still have a lot of moisture in them. And so we saw a lot of them would just drop down to the surface, often stick to a wet surface on the ground and stay there. And they'd stay there really all winter long. And then in the spring, early spring, February and March, the fields would dry out, and that's when the leaves would start moving around. So there was definitely input, but it was delayed. And so if there was any degradation of the BT in the leaves, that may reduce the amount of exposure. So the other side thing that we learned along here is that if you put trees and other riparian vegetation alongside the stream, that can reduce the amount of corn debris going into streams too. So there are ways we can reduce that as well. So our next question is the Cry1AB protein active in senesced corn tissue at harvest? And then is it active after 2 to 8 weeks of environmental exposure? And so the way we did this, Galen Dively had a system that he was using at the time to measure protein bioactivity by using European corn borer fed diet. So he'd take leaf material, include it into the diet of these growing larvae, and the second instars were placed on a treatment diet for 7-day bioassay. And we used a non-BT isoline. One nice thing that we did with our study, for example, was using Streu isolines to try to keep out all of the other effects across varieties. So the non-BT isoline, Cry1AB, and a Cry1AB and Cry3BB1 stacked variety. And then we measure the mean change in mass for each replicate. And so this is designed to be very sensitive to BT to reduce growth without mortality. And we found that at harvest, here's the final larval mass in non-BT, but compared to that Cry1AB and then the stacked Cry1A, AB with the Cry3BB1. And you can see there's a reduction. So that shows that at harvest, there is biological activity of that BT. But when we looked at the leaf tissue, either left on the ground surface like this in little bags like this, or put into the stream in bags and collected over time, we found no more BT activity after 2 weeks of either terrestrial or aquatic environment. So using this Cry1AB in particular, we saw no more activity. So it was no longer active. If that was true, then there's no exposure to invertebrates, so there was no really any risk at all. Um, but at the same time, Monsanto and other companies were putting out new materials, such as Smart Stacks, which included 6 different genes for BT. And so post 2010, we started measuring how long this it was active in the field here. And what we found out was, so the question here then was, do more recent varieties differ in cry protein persistence? And so here's one graph: percent growth inhibition through time with the smart stacks, and you can see that it stays high and significant all the way along up to through past 26 up to 28 weeks. So 28 weeks, that material is still actively affected. So there is BT activity at that point. We sampled a different one as well called Viptera, and you can see the Viptera after 4 to 6 weeks became not so much, so less exposure in that case. And when you look at individual proteins in here, you can see that especially the Cry2A is active throughout the 28-week sequence. So that shows then now we have persistence, and now we were concerned whether or not the stream invertebrates were colonizing the leaves with the cry proteins. So we started to look at that. We had a study where we tried to do 3 things at the same time. We looked at the degradation rates of cry proteins in Senescence corn in water and on land. We quantified the invertebrate colonization of BT and non-BT leaf debris to see if there's a difference in the colonization rates on the 2 different types. And we also looked at the decomposition rates of the leaf debris in an agricultural stream. And so we used these. Well, first of all, we used different sources of the corn debris. There was some hints that maybe different farms, there's going to be a different expression of the BT. So we had 3 source farms with 3 replicate plots for BT corn, as well as the non-BT near isoline. And we collected the leaf material at harvest for experiments. And then in one stream, then we would put out this leaf material either on land or in the water. So these are the bags of the leaf debris. We had 3 source farms. We had 2 different varieties that we were testing. We exposed them in stream and on land. We collected them from 0 to 12 weeks, and we had 3 different reps in the stream that we were collecting from. So what did we find? The degradation using qualitative ELISA approaches, you can see that the positives at 8 weeks, we find Cry1F, Cry2A, Cry34AB, all of those were still positive after 8 weeks. By 12 weeks, only the Cry2 was 100%, but they were still very much out there, the other ones as well. The Cry3BB1 less so at 12 weeks. But when we looked at overall the patterns, there's no clear patterns of difference by source of the leaf debris, that is the farm, or if the debris was in or out of the water, there was still the same level of persistence overall. Quantitative ELISAs we measured as well, and starting with 40 micrograms per gram tissue on with the harvest. It does drop with time, but you can see between 2 and 8 weeks after harvest, there was still significant amounts of BT in that material. We found no significant difference in degradation rates among leaf source or exposure to water versus the land. So those factors were not important. And then invertebrate colonization, BT and non-BT. First of all, we saw no real difference in the colonization on BT versus non-BT leaves. And you can see, okay, the first 2 weeks, you see very little colonization. That's sort of what we expect, because when leaves first go into the stream, they are simply those crackers. the part without any value to them. It's only when the microbes colonize them that they become important food for the shredders. And that occurs after 2 weeks. So the 2 to 4 weeks is when the microorganisms are really colonizing them and growing. And so we see significant amount of numbers of individuals at 4, 8, and growing even to 12 weeks. The decomposition of leaf debris comparing to BT and non-BT, we see very little difference in the rate of decomposition of the debris. If you look out of the stream on the land, the debris tends not to um, uh, decompose very quickly, whereas in the water it will decompose much faster. So it basically disappears over time. But still, between 2 and 8 weeks, you still see significant amount of material in that stream. So, what all this research shows is that the colonizing aquatic invertebrates are definitely exposed to BT. They are finding these leaves, these BT leaves. They are feeding on that material. They are collected on that material and causing decomposition of the leaf material. But we don't know whether they were affected or not for sure. So that led us then down the road to look at the effect. If we do and seeing that we do see exposure now, we wanted to know, are stream invertebrates negatively affected by clay proteins? And we can do this at the individual level, but we can also do it at the ecosystem level. So starting with the individual level, are non-target shredders affected lethally or sublethally? When we first started this work, we did this directly with corn tissue. So we used those near isolines of non-BT, Cry1AB isoline, and then the stacked isoline. And we fed it to that Pycnopsyche caddisfly illustrated here. We also fed it to Lepidostoma, common in agricultural streams. And we found no effect on them. If anything, we actually saw an increase in mass with the Pycnopsyche on the BT isoline. But interestingly enough, so we did this not only with these two caddisflies, but we also tested a cranefly larva species as well as an isopod, aquatic isopod, and they were negatively affected. But only the Cry1AB isoline found a difference in mass, a reduction in mass. For the isopod, we see a reduction in length, reduction in final mass, and a reduction in survival, which was surprising to us. And the reason it's surprising is because we know very well the mechanism for how BT affects their target insects. It's a complex process. It requires solubilization of the BT. It has to be activated, has to bind to the receptor. It will lead to death of the cells, which leads to septicemia overall, and which end up killing the larvae overall. We know this occurs in Lepidoptera and the Coleoptera, the target insects, but this has never been observed in anything like an isopod, for example. So that was not likely to be the case. What else was going on? So I can't remember if I show it here. Yeah, so we found no negative effect on either caddisfly. Both the tupulid as well as the isopod were negatively affected by the Cry1AB leaves compared to the non-BT isoline. However, the response to the stacked isoline leaves did not differ from the BT isoline, which is very strange, right? Okay, so what's going on here? So here's the way I illustrate it is that if you think of a corn seed that goes into the ground in May and planted and it sprouts, that seedling corn, the difference between those two seedlings is that one has BT in it and the other one does not have the BT protein. Uh, as the plants grow through time, the one that's non-BT, it will be attacked by caterpillars, by rootworms, etc. And so they will be attacked by these insect pests, whereas the other one is not. And as at the end of the year, then there's going to be a difference in the tissue because of the insects attack during that year. So that's what we were seeing, we think, on the isopod and the tepulid was a tissue mediated difference, not the presence or absence of the gray proteins, right? So what this work showed us then was that there was a need for a tier one test. We can't use the leaf material to feed them to get a good result on whether or not they are affected by BT. So we started working with Chen Wang in food safety. She was working on nanoparticles. She would put nanoparticles on food items that then we could test. And so we were able to obtain the purified pure protein from Monsanto in a little tiny vial worth about $10,000. And we would put that onto artificial diet for these for the amphipod and then later for the caddisfly. And I'm not going to go to all the details here, but when we fed it to these caddisflies, this nanoparticle, the purified protein, there was no response. There was no sublethal effect. There was no lethal effect. We found nothing significant in its effect to the Pycnosphaera caddisflies. So that satisfied us with the organismal level, but still we wondered at the ecosystem level, could there be harm going on? Are decomposition rates different on BT versus non-BT corn debris? Are colonization rates different? And the way that we did this in the beginning was all in one stream. We'd have BT and non-BT in one stream. That was not truly an ecosystem level test. But we wanted to test this function of decomposition of leaf tissue and the colonization by macroinvertebrates. And to do this, so this is sort of the end result that we were looking for. This is from that Swan et al. Article 2009, looking at the breakdown rate, the decomposition rate versus the invertebrate abundance. You can see that there's a linear relationship there with greater decomposition with more invertebrates involved. But if you look at the individual treatments of the non-BT single gene and stacked, you can see they're scattered throughout. There is no significant effect of treatment on this regression line. We wanted to find out if is there truly no relationship there or not, if the presence of BT leads to a difference in the response of decomposition. And to do this, then we needed large replicates. And so we used different farms, 5 different farms. Here's the illustrated one here. It has 2 ditches. Here's the farm. It's an organic farm. No history of BT corn on this farm. And here's one ditch for water leading down here and out. Here's another ditch over here. And so we treated these 2 ditches as replicates, as individual, not as replicates, as individual for individual treatments. So one would get BT corn and one would get non-BT corn. And then we looked at the effects of the 2. All right. And we did this at 5 different farms that were known to have reduced amount or no BT corn in their history, 4 on the Eastern Shore and 1 near Clarksville. So we had 5 farms, and these farms served as blocks. This is our last experiment here, getting near the end here. This is a backy design. So on each farm, 2 streams or ditches were randomly assigned as either receiving BT post-harvest corn debris added over 5 months, or the near isoline without BT corn debris added at the same time. So we had 2 different treatments. And we did this over 3 years. The first year, we wanted to find out what happens before we add any corn material to it. So we measured structure and function before the debris was added. And then in 2016, we would add the material and looked at the result on structure and function after the debris as well, and then repeat that in 2017. And we measured, measurements include the colonization and decomposition of corn and tree leaf packs. We can either use corn leaves or tree leaves in our leaf packs. And we had, for each of the stream ditches, we had 3 different locations that we served as replicates. All right. So, and here's an example. We're going to grow from this simple chart here. So this is one farm, one experiment, and one measurement. So this shows taxa richness at the Chino farm for looking at tree leaf packs that we put in there. I can't remember if this is in the spring or the fall, but one of those two. But you can see that the non-BT had significantly more in it, more taxa than the BT. But this is before any treatment was added. So the real test, and it is significant, that difference, but the real test is whether or not there's an interaction so that the next year, after you added the BT corn, would you see a reduction in that BT richness in comparison to the non-BT? And you do not see that here. You see a similar amounts all 3 years in this particular case. All right, so that's one farm, one experiment, taxa richness. Now let's jump up to the 5 farms. So now we have all 5 farms. Here's the Chino, and here we have the other farms. And if you look broadly across all of these, we see sort of the same pattern where there's no real significant result on species richness, on taxa richness across these different farms, with or without BT. In the leaf debris. All right, so now we can go, this is 5 farms, one experiment, and taxa richness only. Now we can go all the way up to, now we have 3 different experiments, the corn leaves, the tree leaves in the spring, tree leaves in the fall, across 7 different measurements, breakdown rate, abundance, Shannon evenness, functional diversity, taxa richness, Shannon diversity, Hilsenhoff index, even we included. And this is non-BT versus BT here. If you look across all of this, we see no significant interaction except for one place here for the Shannon evenness, for the tree leaves, where we looked at the experiment in the spring. You can see that going from the first year to the second and third, you see an increase in the non-BT leaves, an increase in evenness, whereas we see a decline in the evenness going from the first year to this second and third year. So that was the only significant one. We did a deep dive on the data here. There is nothing about caddisflies in here that was going on. There was nothing about anything that suggested that there's anything real about this effect. You would expect across this many replicates and this much data, basically you would see some significant effect someplace. So this was our one significant effect that we found. So, and which really was not much of an alarm to us at all. But going back then, we also were interested in this relationship, breakdown rate versus invertebrate abundance. And like I mentioned before, when we first did this was in one stream. We were not doing this at the ecosystem level, but we were able to do this at the ecosystem level with this other one. And so here we show the tree leaf packs in the fall. The blue markers are those that are receiving BT corn versus the orange that are receiving non-BT corn. You see this relationship between breakdown rate and abundance. The slope is the same for the blue versus the orange. The intercept is different, but there's no real reason to think that there's any effect specifically on BT on this data. Same thing, well, here we see all of it jump together so that even though we see this same pattern of breakdown rate versus abundance, there is no effect of BT versus the non-BT in tree leaf packs in the spring. And then corn leaf packs in the spring, the same thing. No, there's a relationship, breakdown rate versus abundance, but there's no difference between them. All right. So from all of this, the results were that no changes in the relationship between breakdown rate and macroinvertebrate abundance between BT and non-BT corn isolines added to streams and ditches overall. And so our conclusions from this experiment, which we still are working on the manuscript right now, hope to get that out soon. No negative effects in factors measured due to the addition of BT corn. While the exposure is present from our other data, the effect is negligible, so risk is going to be small. This corroborates other work within the lab. It also indicates minimal risk in terms of effects. And this is great news, we think, for farms because BT allows the minimization of pesticide spray and has shown decreased pest pressure in adjacent fields, further decreasing the needs for pesticide applications. So good things for BT. All right. So in conclusion, environmental risk is a function of exposure and hazard effect. And we know that we have BT in this corn material, which goes into the stream. It stays active in the streams during the time when things like this caddisfly, Pycnosphaera caddisfly, is colonizing those leaves and feeding on those leaves. It is breaking down quickly in the stream, but not quickly enough to not be an exposure to the invertebrates. So the point is that we need to focus our research on non-target effects. We have exposure for sure. And when we did that, we have seen no evidence of any effect. Now, the current climate is changing. Of course, we have increasing number of genes, including new RNAi technology, which is not being tested hardly at all anymore. Not too surprisingly, given the politics. Increasing lack of non-BT isolines. We depend upon those non-BT isolines to do a lot of our work. You can't get many of those anymore from places like Monsanto. This still remains highly controversial. Shoppers will avoid eating GMO corn material, so food products. Persistence of BT protein in senesced tissue suggests that risk assessment should focus on minimizing non-target effects. So anything we can do to reduce that corn debris going into the stream would be good. And the current best practice is to test individual species for acute chronic effects and to not test effects in the field at community ecosystem levels. So it's going to be someone other than the government who looks at ecosystem level effects because the government will focus on individual species for looking for acute and chronic effects in risk assessment. So here's my conclusion for the overall story here. So here's a picture of Maryland cornfield in May. If you look close, you can see here's the new corn growing here, but there's corn debris left over from the year before. We know now that this corn debris, even though it's been there for over a half a year, it still has BT in it. It will go into streams. It will expose aquatic invertebrates, but we see no effect in our studies on either the individual level or at the ecosystem level. So is BT corn a risk to stream ecosystems? I would say that the data says no. But this is from Beck. Neither can we prove by experiments or simulations what people have to accept, nor can any risk calculations be established by technical or bureaucratic dictatorship. Risk calculations require what they are supposed to generate, cultural acceptance. Ultimately, an acceptable risk is an accepted risk. And that's the issue. We have people that are concerned about GMO crops, and yet there's no reason to think so. There are positive things about them in terms of our environment, reducing pesticides, but Nevertheless, the public still has not uh, is still concerned about GMO in our food supply. With that, I'll end with acknowledgments. This shows the LAMP Lab back in 2014 when we were actively doing a lot of this work. Now we are done, and our work is more on farm biodiversity and wetland ecology. We were supported by the BRAG program, 3 different rounds to do this work. A lot of people were involved, and I thank them all for that. And with that, I conclude and I will stop sharing. So, uh, are there any questions? I haven't looked at the chat, of course, at all. So, these are the people setting in. Any questions for me?
Oh, I have one.
Thank you, Fred. Yes.
Sure. Uh, Great presentation. Thank you so much for all the information there. Um, I was curious, this all began because of the paper that was published back when that they were claiming that there had been some deleterious effects, right, on the stream insects. And
Right.
it spawned all this, it fostered all this research, right? And what were they basing this on back then? I mean, what did they have something that they really found that they thought they had? And apparently they did. So,
Well, it's been a long time since I've looked at the paper, but for example, they did aquarium studies with filter feeding caddisflies and put BT pollen into those streams and those artificial streams, and they found at very high rates of pollen, way exceeding anything you'd see in nature. They
right.
found a negative effect on the caddisfly. So if you overfeed a caddisfly, it will not do very well. Things like that. And from that kind of an experience, that's what they put this generalization about widespread impacts on our stream ecosystems. So it was called out, and so it led to this research, and since then, the USDA is satisfied that it is a safe thing to use and so forth, not deleterious to the environment. And most people, most of the public have accepted, I would say, generally, but there's certainly a lot of people that are still concerned about GM crops.
Oh yeah, absolutely. Um, you know, this is completely exactly like the monarch study that they did where they fed the BT leaves to the monarchs and oh wow, it's killing them or that they dusted the milkweed with BT and basically said, hey, it killed the monarchs. Um, I'm just amazed at the parallels of this, what you've just described. Didn't they learn anything from that? I wonder.
Well, what can you say? I mean,
Yeah.
I mean, what John Losi did was a valid experiment.
Hmm.
If he were to put that in the light of if there was exposure, then there could be this effect. But he didn't do that. I mean, he just basically said that there could be, a lot of death of monarch butterflies because they are in the area of these crops. So,
Pretty amazing. Yeah. They anyway. Anybody else have some questions that they would like to ask Dr. Lamp? He covered a lot of stuff. Um, so feel free to turn your microphone on and ask a question if you'd like. Let me check the chat and see if there's anything there. I don't see a question. Wait, there's some messages down below here. Hold on a second. All right.
Harold asks about other countries. It's really interesting that it's very spotty. There are some countries that have taken advantage of the BT gene, and others that have not. And they are very serious about it in Europe, and they're very careful about that. So they're doing the science, and they do have certain crops that are genetically modified. Um, but if you think about it, it would be so much safer if you think about what you see in terms of the spraying of hard pesticides in some of these crops in places like India. If you used BT to kill the caterpillars that are feeding on certain crops there, uh, instead of spraying those pesticides, you'd have much less of impact in terms of the health of the people and the environment. And so, uh, yeah, so there's good reason to use it when you can. Um, I saw a question there for just a second. Um, what do they misunderstand? Well, I guess when they first think back to when they first started doing this, and they were putting these genes from bacteria into plants, and you just had this sense that this is probably a wrong thing to do, right? And so I think that basically that feeling sticks with you. You don't look at the science, you just have that feeling that there's a problem. When I give these talks and talk to people and talk to them specifically about the reasons why they don't like the concept of genetically modified crops, they say it's Monsanto. They say they don't like Monsanto making all this money from BT corn. And I say, well, that's capitalism. They invested a lot of time and effort into producing this crop, and they uh, they need to get their money back, and that's the way capitalism works. Uh, and it has nothing to do with, and it's not causing harm. It's actually causing less harm to the environment than the choices in many cases. So, uh, yeah, so it's a misunderstanding, I would say. Let's see. How does the risk GMO mosquitoes? Well, that's a really good question. I'm not in that world anymore, so I can't really answer that. But you have to be very careful when you start introducing insects that are uh, like GMO mosquitoes. I know that my colleague Ray St. Ledger is doing some of that work, and so that's a good question for him someday. Any other questions?
But you were mentioning about Europe, and they're cautious about growing BT corn and whatnot. Is it the same in South America, or are they a little more liberal about it down there? Do you know?
I don't know all across, I know a few countries basically what's going on, but generally speaking, most places are interested in looking at it and considering the possibilities. They're looking at the safety factor both to people as well as to the environment. Um, and so if they get enough from their business in that particular country, they will accept it and use it. Yeah.
Anybody else have any question they would like to ask Dr. Lamp? I have one other one if nobody else chimes in. So we talked about tonight, or you talked about the effects, and I think you mentioned up to 6 months, right? You said, I was just wondering, do we know what the threshold is for overall the lasting effect of BT? How much time can that be active uh, in the environment? A year or more, or what?
Well, the leaf material itself is degrading at the same time. And
Yeah.
once the leaf debris is completely done, as far as we know, there's not much BT left either. Basically, as you lose the material over time, and generally, if you think about it, in spring, there's a lot of degradation. It's very rapid, the degradation of the leaf material. And so most of the BT is going to disappear at that point, one way or the other, or you couldn't probably measure it overall. So,
So it
yeah,
degrades along with the vegetation. So that
yeah,
includes the corn cobs and the stalks, all that as well. Then,
yeah.
okay. There's one other question popped up on the chat.
Oh,
What?
let's see.
Hmm.
Uh, well, I'm not sure what all the factors are, why other countries are hesitant to use and produce GMO crops. Um, Certainly, it's a more expensive approach than their standard things that they do. So that could be a big part of it. Um, but and they probably would want to do a lot of their own safety testing and things like that as well. I'm not sure. I mean, that's a little bit beyond my expertise overall.
Maybe there's sometimes the GMOs get a bad rap, and I think,
Well, they certainly get a bad
yeah,
rap sometimes.
yeah, that plays a role. Unfortunately, there's a lot of good in it. I try to always tell my students about the benefits of various GMOs
Yeah,
and
yeah,
how, they produce things that help humankind, So anyway, um, I guess we have to see how things pan out in the future with all this. Um,
yeah. There's
it's
another
interesting
question
too.
there from
Yeah.
Harold on
Uh-huh.
liquid feeding critters. Well, they do know that they do have BT products now that will be picked up by aphids and affect aphids and leafhoppers. I'm not sure where they are in the development chain.
Mm.
I don't think there are any products out yet, but they are working on those kinds of products out there right now. There's a lot of activity, especially since we're starting to lose some of the uh, um, efficacy of BT crops because of the resistance that has developed to it.
Well, that
Uh,
might be an interesting question too. I mean, because what is
yeah,
the resistance like
yeah,
on
it's
these?
it's developing. There's development of resistance, especially corn earworm has become resistant to BT. So interestingly enough, not so much the corn earworm, but the other ones.
Even with the stacked genes where they're putting six different genes in there?
Yeah, yeah,
Wow. Oh, wow.
yeah.
Yeah,
Yeah.
that's crazy.
Before I go, though, I do want to mention again about EntoQuest. I think that this is a great opportunity for those of you who love insects. And if you like to serve as educators, if you've got young people in your area that are going to school or whatever, it's a great place to bring young people to. We even have high school students that come to EntoQuest. And for a weekend, we just go out and collect and have workshops, a lot of different activities to help them. We've had visitors, we've had attendees from Texas and Tennessee and Florida come up to the Eastern Branch to attend these things. We've had University of Virginia paid for the registration for like six of their students to attend the meeting this last year. So it's a good opportunity for undergraduates in particular, I think, but also new graduate students would gain from this. We have a lot of then we have adults that come along and they just provide their expertise along the way. And if you've got an idea for a workshop or a collecting adventure at one of these places, volunteer. We'd love to have you there. And watch the information at the ESA website, and maybe we can see some of you at the Pennsylvania meeting next September.
Does EntoQuest have their own website?
They are part of the Eastern Branch. So if you go to the
Okay.
Eastern Branch ESA, They
Yeah,
have a link there to past meetings.
great. Okay. All right. Well, Dr. Lamb, thank you so much
You're
for
so welcome.
presenting tonight. We really appreciate that. Sorry for the little delay beforehand, but I'm glad it all went off well.
Yeah, thank
Um,
you.
I was doing a little damage assessment and kind of like got away from me. Um, but anyway, this was nice, and I hope we see you again sometime in the future. So good luck with the continued work and your research. Um,
We've liked to publish in your journal. We've had
oh
three
yeah,
articles, I think, in the journal, so that's good.
great. Yeah, please. Uh, Gene Scarpilla does a
I
fabulous
love I
job.
love hearing comments like that.
Absolutely. Yeah, yeah. Gene is fabulous with the editorship of that journal. So if there are other people that want to come forward, grad students, any kind of publications like that are more than welcome. So we thank you. So I was just curious that you've been at College Park for quite a long time.
I
Are
have.
you emeritus at this point, or are you still?
I I have one last grant that I'm working on. That finishes next summer. I hope to graduate. I mean, graduate. Hope to retire fully. I'm half retired now. I'll fully retire a year from now, and I've got one graduate student left now. So I'm winding down quickly. Yeah. No, I've been there for 40 years. So yeah,
Wow, that's a
yeah,
long time. Yeah, well, I've been at it for 33 years, so I understand. But 40,
yeah,
that's a milestone. Well, that's wonderful. But the nice thing about is in academia, you can continue as long as you wish to, basically, and things are going well and people do care. And you said Galen Dively is still there too, right?
yeah, 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 yeah.
Is
He's
he?
still coming in every once in a while. Not as much as he used to, but still comes
uh
in.
That's great. And I know Mike does as well, Mike Ropp. So
Yeah.
um, he'll be speaking for us in March.
I
So
saw that. Yeah, that's
yeah,
very good.
yeah, and it's great. And he has some pretty cool things to share, which could be rather sobering, actually. So
Yeah.